Hello students, today I will perform a slide for pollen grain germination. So let's see the requirement. You required Vinca flower. So this is Vinca flower. In Marathi we called Sadafuli. It flowers throughout the year. Next is sucrose solution. I've made this sucrose solution by taking 10 gram of sucrose in 100 ml of distilled water, plus I've also taken 10 milligram of boric acid. Boric acid fastens the germination. If the pollen grain burst, kindly decrease the sucrose concentration. You can take 5 gram of sucrose in 100 ml of distilled water. Next requirement is safranin. Safranin stains the lignified cells. Then forceps, needle, slide and cover slip. Very important, even microscope is required. So let's start with the procedure. You can see this is Vinca flower. Remove the petals one by one. So petals are removed. Anthers are present near the mouth of the corolla tube. This is pedicel. On the upper side, there is a corolla tube. So over there, anthers are present. And for that, you have to open the pedicel part. First of all, I will take sucrose solution on the slide. Take adequate amount of sucrose solution. So I've taken sucrose solution on the slide. Now with the help of needle, open the pedicel part, the upper side. Over there you find anthers. Dip the part into the sucrose so the pollen grains will come out. Follow this procedure. So the pollen grains get transferred into the sucrose solution. The pollen grains are microscopic. You have to keep this for 10 to 15 minutes. Now you also take little amount of safranin. Safranin stains the lignified cells. So I am taking 1-2 drops of safranin. You have to keep this for around 10 to 15 minutes. And then you will place the cover slip. So now I am placing the cover slip. Hold the cover slip with the help of forceps, and another end of cover slip should touch the slide. So see the procedure. One side of the cover slip should touch the slide and slowly remove the forceps. If there are any air bubbles, with the help of needle, lift the cover slip and again place it down. When you place the cover slip, excess of sucrose solution comes out. Remove it with the help of blotting paper. See that your slide is clean. So now slide is ready. Now coming to adjustment of microscope. This is compound microscope. This is a condenser. To adjust the diaphragm, now adjust the mirror. So the light will be focused on the aperture. This is aperture. To adjust the mirror, so you will see light in the aperture. And we are adjusting under low power, that is 10x. So keep the slide. Keep minimum distance between the object and the objective. And then move the body tube in upward direction. So now I am adjusting the body tube. So now slide is adjusted. Now let's see through the eyepiece. So now you will see the pollen grains. These are normal pollen grains. Germination has not started. So we'll see the pollen grains with exine and intine. Since it's a dicot, there are 3 germ pores. After some time, you will see the pollen tube. So we can see the pollen tube. So germination has taken place. This is the pollen tube. I had made one more slide without using safranine. So you can see over here. This is the pollen tube. When pollen grains are kept in sucrose solution, the intine comes out through the germ pore and forms pollen tube.